All right. So when we left off on Thursday, we were talking about translation, and I had shown you a slide like this exact slide, in fact, that covers a couple of important concepts. So one important concept is this idea of that degeneracy of the amino acid code, that the cell can tolerate some mutations and still get the same amino acid. So that's really beneficial because we get a lot of mutations all day, every time the cell replicates, every time we're exposed to some sort of chemical. And so the ability for our bodies to tolerate some of those mutations and give us the same amino acid really protects the integrity of the cell's development. So our cells, bacterial cells, etc. The other thing we talked about was the tRNA, which is one of those types of RNA that's really important for bringing amino acids to the ribosome for the process of translation. The tRNA knows, well, it doesn't know anything, right? But the tRNA goes where it goes, and you get this interaction to occur because tRNAs will have a region of the molecule itself that's called the anticodon. So it is complementary to the codon, which allows the codon and the anticodon to interact so that we can have amino acid binding. So what that looks like is, well, translation. With translation, we're looking at the ribosome. Remember, in prokaryotes, the ribosomes are all free-floating within the cytoplasm. In eukaryotes, there are ribosomes that free-float in the cytoplasm, but we often talk about ribosomes that are associated with what structure? The ER, right? The endoplasmic reticulum is studded with ribosomes, at least the rough ER. So there's protein synthesis that can occur in the cytoplasm, and there's protein synthesis that can occur in the ER. So with translation, in order to get translation to occur, you have to get the small and large ribosomal subunits together with the mRNA and the first tRNA that brings in the first amino acid. So how does the ribosome recognize that there's a piece of RNA that it needs to interact with, that there's a piece of mRNA? Well, it recognizes a region on the mRNA that's called the ribosome binding sequence. So when we're talking about this process of transcription and translation, sometimes when we think about it, we think, well, the first nucleotide that's going to be transcribed is going to be for the start codon. You're going to get AUG. That's not how transcription works. So, you'll have base number 1, right? But maybe down in here is somewhere where you'll eventually, right, transcribe it into AUG. So there's going to be, if we're looking at the mRNA transcript, if you're looking at your mRNA transcript, you have your 5' end, which is the front. You have some number of nucleotides. Then you'll have AUG, some number of nucleotides, a stop codon, and more nucleotides. We don't transcribe right at the beginning of what we need, because we need this part right here. But we transcribe all of this. So this is the piece of RNA that we have. This region up here is called the 5' untranslated region. It will not give us a protein, but it does exist as part of the RNA. And down here, after the stop codon, we have the 3' untranslated region. It isn't going to give us a protein, but it does exist as part of the RNA. And so within this 5' untranslated region, somewhere in there is going to be the ribosome binding sequence. So the ribosome comes together and still has space to work. It doesn't have to start work like right there. When everything finally comes together, it looks something like this. So you have the small and the large subunits are now together. The mRNA is right in there. And now you have these pockets on the ribosome. And if we look at the pockets from left to right, the EPA. I always remember them as exit, peptide, and then the amino acyl. That's what it's called. So EPA, left to right. So that first tRNA, right, has an anticodon that allows it to bind to the first codon. And look, you have this untranslated region hanging out here. So you have your mRNA, here's your first codon, AUG. Your first tRNA comes in, and the anticodon is complementary to the codon, so you have an interaction. And it's got methionine. The first amino acid is always methionine, or a methionine derivative. So that's in the P site. E site is empty, A site is empty. But now what can happen is the second tRNA recognizes the codon that's available in the A site. The codon is UUA. So the anticodon that comes in has to be AAU, because that's the complementary interaction. And in this case, it's happening to bring in leucine. Okay, cool, whatever. It doesn't really matter what comes second. As long as methionine comes first, whatever else can come second. Doesn't matter. But now what happens? Now you have your two tRNAs. You've got your two amino acids. How do we get the protein? Well, we start making the protein because this methionine, right, that's in the P site will join to the leucine that's in the A site. They will form a peptide bond. It's a covalent bond. It's a very strong bond. It requires a lot of energy. And again, this process is taking place like milliseconds. This is not a slow process. So now things keep moving, right? The cell doesn't stop making proteins, it just keeps moving. So the ribosome continues to move along the mRNA. Now we have AUG in the E site with an empty tRNA. Because remember, the methionine jumped over to join the leucine. We have our peptide chain here in the P site, and we have the new tRNA that came into the A site. So what happens now is the empty tRNA is released. It can be reused, it can be recharged. So another methionine can be added back onto this. Now, this bond here between the tRNA and the leucine is cleaved. These two amino acids will be joined to the glycine. And then we scoot over again. And we scoot over again. Right? We just keep scooting over. The process just repeats. 100, 200, 300, however long the protein is, it repeats that many times. Until in the P site, here's my favorite stop codon, UGA. There is no tRNA, so nothing comes in. UGA signals to the ribosome that protein synthesis is over, we're done. There's nothing else that's going to happen. So when there's a codon present in the A site, but no tRNA comes in, the protein is released and everything falls apart. The mRNA could be reused as long as it's still good. The ribosomes can come back together and synthesize another protein. All of this can be recycled for a little while. The mRNA is going to be the least stable thing of all of it. Everything else is a lot more stable. The mRNA will eventually degrade. It has a very short half-life. But theoretically, right, we could reuse it until it's not usable anymore. So we can replicate our DNA, we transcribe it into various kinds of RNA, and then we use a lot of those types of RNA, right? We use our RNA to help build the ribosomes, we use tRNA to bring in amino acids, and then of course mRNA is the big one that we use as the template for protein synthesis. And broadly, these things are conserved among prokaryotes and eukaryotes. So if you haven't taken genetics yet, you understand this. Humans are not that different. We make it a little more complicated, but the broad processes are very, very similar. So what do bacteria do that makes it different? Not just that it's simpler. We don't have introns and exons, right? We don't have lots of versions of some of these proteins. One of the things that makes bacteria so efficient is the fact that they can do what is called simultaneous transcription and translation. So what they're trying to illustrate in both the cartoon and in the actual transmission electron micrograph is that you can have a cell that is making an RNA, so it's making the RNA, making the mRNA, and as soon as that ribosome binding sequence is available, theoretically a ribosome can come and attach and start translating the RNA into a protein. So here's our DNA template. Here's our various RNAs, right? Because these are our RNA polymerases. So it's made a long RNA, a little bit shorter, pretty short because it's just started. Maybe another one pops on and starts transcribing again. And you have all of these ribosomes that latch on and start making many, many copies of the protein all at the same time. So bacteria are very efficient and they can very quickly react to changes in their environment, because as soon as they start making a new RNA, they can make tons of copies of that protein immediately before the RNA is even finished. And so when you have the multiple ribosomes on the same transcript, we call that polyribosomes. So again, they've tried to illustrate it here in this cartoon, and they're trying to illustrate it with this transmission electron micrograph as well. Eukaryotes are not capable of doing this for two major reasons. Anyone want to hazard a guess as to one or both of those reasons?
It might need to be post-translational modifications for this.
Yeah, post-transcriptional. Yeah, post-transcriptional, exactly. We have to, as eukaryotes, we modify our RNA really significantly before we start translation. You might remember that we have to splice out introns, so pieces of DNA that won't give us a protein, which means we have to join together the exons. We also add what's called the 5' cap. So on the 5' end of the transcript, we add a modified base. It's our 5' cap. And we add a 3' poly A tail. We add, 100, 200 adenines at the very end because, again, RNA is pretty unstable, especially because it is single-stranded. It's very prone to being degraded. And the easiest way that these enzymes that degrade DNA, they work from the 3' end, and they just start pulling the RNA apart. So if they have to deal with 200 A's before they actually start affecting the transcript itself, that's very helpful for us. So yes, one, we have complicated the process by having to process our RNA so much. What is the other major reason?
The nucleus, the mRNA has to go from the nucleus to the cell.
Yeah. Ribosomes don't exist in the nucleus. So even if we didn't have to process our RNA so much, we have physically excluded the protein synthesis machinery from the transcript access. And that's why we have to process our RNA so much, is because we have to export it. So it has to be stable enough for us to export, get to the ribosome, and then do translation. If you ignore introns. So we have a more complex genome in terms of having to remove the introns and glue back together the exons. We also modify the RNA by adding the 5' cap and the 3' poly A tail, but then we have physically separated these two processes. They are spatially separated in the cell. But because prokaryotes don't have that type of spatial separation for really anything, they can do these processes at the same time. So sometimes, evolution didn't go the most efficient route. Because I would argue this is far more efficient than what our cells do. Okay, so kind of in summary, right, you'll have a promoter, which is the region of DNA that is recognized by the RNA polymerase to allow transcription to occur. So RNA polymerase is the enzyme that makes the mRNA. Within the mRNA, in the 5' untranslated region, this 5' UTR, you will have a ribosome binding site. The ribosome will bind there and can begin translation when it reaches the AUG. There's another term on this slide that we are going to come to in just a minute. Polycistronic mRNA. We will come back to that. But broadly, right, you have the transcription and the translation that gives you the proteins. So what questions do you have about replication, transcription, or translation? And as always, shoot me an email, come to office hours. Okay. All cells have to regulate gene expression. We're going to focus on how, of course, bacterial cells do it. But what I mean by that is, it's kind of the basis behind the same phenomenon. All of our cells in our body, right, as, multicellular eukaryotic organisms, with the exception of our red blood cells, have the same DNA. Anyone disagree? All okay with that? Your liver has the same DNA as your eyeballs, as your kidneys, as your skin. We're good? Okay. Why are all of those cells so different? They all have the same blueprint. Different differentiation. What controls that? Why would a cell differentiate? Why do I have an eyeball cell and a liver cell and a skin cell? I've got the same instructions, got the same book. Maybe I read different chapters, right? If we talk about it as a book. Or you have a blueprint to a city. Do you build all of the city buildings all at one time? Well, no, maybe you're going to build the school today. And within the school, you have classrooms and lab rooms and a cafeteria and a library. You don't read every page. You should of this book, but other books, maybe not. So in our bodies, right, the genes that our cells use are different. We have all the same genes in all of the cells, but only the genes that the liver cell needs are on in the liver. That's what led to that differentiation. My eyeball cells, all the different cells in my eyeball, right? The rods and the cones and the cornea and your lens and blah. They all read slightly different instructions. But within each cell, even in microorganisms, there's a subset of genes that we call constitutive. They're always on. Sometimes we call them housekeeping genes. You have to have a baseline of genes to help you make nucleotides, right? The proteins for nucleic acid biosynthesis or ribosomal synthesis or whatever the cell needs to function. There's a baseline. And in some species, that can be the majority of their genome. In some, it could be less. It depends on the species. In our cells, there are going to be certain types of genes that are on in every or almost every cell type. They're constitutive, they're housekeeping. All of our cells have to do respiration. If a cell doesn't generate energy, it's going to die. But just like in our cells, in bacterial cells, there are genes that they don't need all of the time. And so they can control the expression of those genes. We call them regulated. And we have two broad types of regulation. We have genes that are inducible. So an inducible gene is one that is off almost all the time, but you can turn it on when you need it. And so if we think of the cell like a house, there's, I'm sure, things in your house that you don't use all the time. Like your dishwasher, if you have a dishwasher, it doesn't run 24/7. Or your laundry doesn't run 24/7. It feels like it. If you have kids, it feels like your laundry runs 24/7, but it doesn't. Maybe your TV, right? It doesn't run 24/7. It's usually off, but when you're home and vegging out, you can turn on your TV. Then you have repressible genes. These are genes that are almost always on, but there might be some exception where sometimes we turn it off. Maybe that's your TV. Maybe it's always on and then sometimes you turn it off, but not very commonly. Or, maybe it's the internet to your house. Maybe you turn it off sometimes if, you have to reset your router, or you turn off the water because you've got to do something with the plumbing, right? Things that are normally on, but there might be an exception where you can turn it off. And then we'll talk about catabolite repression in the context of that in just a little bit. So the idea is every cell, bacterial, eukaryotic, makes a decision, and I hate to, anthropomorphize cells, but that's kind of what happens. It's a molecular decision whether or not to make a gene product. Okay, that is expression. And so we can have a number of different levels of control of gene expression. Again, when we talk about eukaryotes, the number of levels, the number of checkpoints we have is a lot more than bacteria. But we can always have this level of what we call pre-transcriptional control. So, before transcription, the cell commits to either transcribing or not transcribing a gene or set of genes. With repression, we're turning them off. We inhibit gene expression, we decrease enzyme synthesis. For whatever reason, we don't need the product, so we're turning it off. We use proteins called repressors to do so. But by default, these are the ones that are almost always on. We can turn them off if we have to. We can use repressors to turn them off. Normally, they're going to be on. Of course, the other option is to induce gene expression. We can turn it on if it's normally off, like flip the switch, turn on the light. So the default position with inducible is off, but it can be turned on when necessary. Questions about repression and induction? Again, I'm hoping that a lot of this is review. It might not be fresh review. And I might be dragging you kicking and screaming into the review, but that's okay. We suffer together in microbiology. Okay. So the last new term that I want to introduce right now ties into that polycistronic mRNA thing that I brought up a little earlier. Bacteria are really cool. We already talked about how efficient they are, that they can do transcription and translation at the same time. How cool is that? But not only are they that efficient that they can do transcription and translation at the same time, they also have really well organized genomes. Not for every gene, but for many. So there are many areas in a bacterial chromosome where the organism has arranged the genes of a pathway to be right next to each other. It makes sense, right? If you need 5 enzymes, let's put the 5 genes right next to each other. We are like, oh, I need 5 enzymes. I'm gonna put one on chromosome 1, one on chromosome 7, over here on chromosome 14, evolution. Bacteria often put them together. And we call that an operon. So an operon is going to consist of, and we'll see it on the next slide in an image, but a region that is the operator, a region that is the promoter, and then the genes that they control. And so what I mean by that is, as you see here, you've got, I'll draw it out so it stays on the board. So you've got your promoter, which is the region of DNA. What happens at the promoter? In the context of transcription, what happens at the promoter? It's where it starts, because that's where the ribosome will bind, right? So, or not the ribosome, I'm sorry, the polymerase. RNA polymerase binds. Good. Okay. Directly next to it is a region we haven't talked about yet that's called the operator. This is where we'll get some type of repression happening. And then connected to it are going to be any number of structural genes. Here they've shown 5 or 3. This is the classic operon. The Lac Operon is it's the Lac Operon. You can't escape it. I'm still talking about it 20 years later. Right? It's the Lac Operon. So these are your structural genes. The other operon that we classically talk about, and we will talk about, is the Trip Operon. It has 5 structural genes, whatever, 3, 5, doesn't matter. Okay. So somewhere else, right, maybe a little bit farther away, you'll have the I gene. This will be where you end up this makes the repressor. So this produces a protein that we will call the repressor. Okay, so what's the big deal? So they put all these the genes next to each other. Ooh, bacteria, ooh, so awesome. Well, what they did was not just put them together, which is still cool and more efficient than what we did, but they also put them under the control of one promoter. I've got 3 genes, 1 promoter. Normally, 3 genes should have 3 promoters. But if you think about it logically, if I need one of these genes, I probably need all of them. They're part of a pathway. They're part of doing a thing. I don't need 1/3 of the thing, I need the whole thing. So I need to make all of them. Why don't I just turn them all on? Like when you get home and it's dark and you have to find all the light switches. How annoying is that? I want the one light switch that turns on all the lights. Because then I don't have to go into every dark room and be creeped out. I don't have a master light control at my house, so I have to go turn on all the lights. Or turn them all off, right? That's super annoying. What if I could just turn on all the lights I wanted? I can make all the protein I want. And I have them all together. So broadly, every operon will look like this. Maybe a different number of genes, but the organization of the operon itself, right? This whole thing is the operon. There is going to be a promoter, an operator, and then the number of structural genes, often 3 to 5. And maybe a little bit farther away, or maybe very close, it depends on the system, you'll have your repressor. The repressor is not under the control of the operator, right? That's why I put it farther away. It's not under the control of the promoter, it's not under the control of the operator, it is its own independent unit that will help control this. So what this does, if I only have one promoter, how do I make all of these transcripts? Because I can't start at Y and I can't start at A, I can only start in front of Z. Well, what the promoter does, there's no termination signal until the very end of A. So when you start to get transcription, you actually get transcription of a giant mRNA that will have the A region, it'll have the Y region, it'll have Z, it'll have this 5' untranslated region. So it's one mRNA that could, in theory, right, it encodes for 3 proteins. This is the polycistronic mRNA. The mRNA that results from transcription of an operon is a polycistronic mRNA. A cistron is another word for a transcript. So it's a polycistronic mRNA, has multiple transcripts in one. And so again, the best studied operon is the Lac Operon, and so that's the one we'll talk about first. I'm hoping, again, that you've seen this in other classes, so I hope it's review. Maybe the concepts become a little bit more clear. The Lac Operon is our really classic example of, like I said, an inducible operon. It is one that is normally off, but it can be turned on. So why do cells use the Lac Operon? The Lac Operon contains the enzymes necessary for breaking down lactose. Lactose is a disaccharide that consists of glucose joined to galactose. The cell prefers to use glucose as the energy source, so it has to break the lactose to access the glucose. It can modify galactose if it wants to use that as well. But we need to be able to basically import and break down the lactose. So lactose is not maybe a common sugar that the microorganism might encounter. So typically, and it's not the preferred sugar, it prefers glucose. So typically this operon is off. So that works by the repressor protein just sitting here and I can spell on top of the operator. Does that stop RNA polymerase from binding? No. So promoter. RNA polymerase is happy to come in here. I'll just make it RNA polymerase. And then bump into the repressor. It gets nowhere. The repressor is physically blocking transcription. So the operon is off because the repressor is bound. What happens when we have lactose? It's going to get more complicated than this, I apologize, but it does. But in a simple situation, what happens now the cell has lactose? I need to use my lactose. So when lactose gets into the cell, it can be slightly modified into this little molecule called allolactose. So allolactose is the inducer, so I'm going to make it blue. So this is the inducer. And this is how any inducible operon would work. There's going to be an inducer. Okay. But we've already talked about this. What happens when you shove something onto a protein? It changes shape. You shove an inducer onto the protein, it changes shape. And so now it's down here, bound to its little inducer. And here goes RNA polymerase. So now RNA polymerase can transcribe. So because we have that inducer, it binds to the repressor, causing the repressor to fall off. Eventually the cell will use up all of the lactose, okay, meaning bye-bye. And then we come back and block again. So that's the simplified version of the Lac Operon, or any inducible operon. Normally off, but when the product that we can use is available, we can induce transcription of the operon. Notice the inducer does not bind to the DNA. The inducer only binds to the repressor protein. Repressor proteins are what interact with DNA. The inducer is going to be a small molecule that interacts with the repressor protein itself. Doesn't touch the DNA. Okay. I like animations because they're sort of like movies, but not really. So we have a little animation as well. And let's see here. These are from a different textbook, so that's why I put them in the slides for you.
E. coli includes a promoter, an operator, and 3 structural genes encoding proteins involved in the catabolism of lactose. The Lac Operon is an inducible operon, meaning that the genes are usually not transcribed. When lactose is available to the cell, the operon is induced or stimulated to produce the proteins of lactose catabolism. The E. coli chromosome contains a regulatory gene near the Lac Operon. This gene is constantly transcribed and translated to produce repressor protein. The repressor protein binds to the operator DNA at the Lac operator, physically preventing RNA polymerase from moving beyond the promoter. When a repressor protein is bound, the genes for lactose catabolism cannot be transcribed into mRNA. When lactose is present in the cell, some lactose molecules are converted to allolactose. Allolactose binds to and inactivates the repressor proteins, preventing them from binding to the operator. The structural genes are transcribed, resulting in the production of the enzymes needed to catabolize lactose. Since allolactose induces or stimulates transcription of the Lac Operon, it is called an inducer. Notice that the inducer acts by turning off the repressor, not by actively stimulating the genes to be transcribed. When lactose is not available, the repressor proteins are active. The structural genes of the Lac Operon are not transcribed. This control prevents the cell from producing the proteins for lactose catabolism when lactose is not available.
And so that's a really important thing for cells to be able to do, is to be able to control when they make transcripts and therefore when they make proteins. It costs 1 ATP per base to transcribe. It costs 2 ATP per amino acid to translate, 1 to move the ribosome, 1 to catalyze the peptide bond formation. It is expensive to do transcription and translation, especially when you don't need to. So keeping a gene off for maybe a more rare metabolite you want to process is a really beneficial evolutionary strategy. We infrequently encounter lactose. We infrequently turn on the genes necessary to break that lactose down. questions about an inducible operon?
Is
Yes.
this like the direct pathway responsible for giving you more lactose tolerance when you like chug more milk or something?
Mm-mm. That's a human enzyme. It's a human enzyme that's responsible for lactose tolerance.
This is only for bacteria.
Only for bacteria, yeah. I mean, you know, those of you who are taking the lab this semester, this is one of the biochemical tests you'll perform. You'll test for the ability for some of the microbes to ferment lactose. Not all of them can do it because not all of them have these enzymes that allow them to break down lactose, but E. coli should. In theory, when you're testing E. coli, that one should be positive. If not, that's okay, but it should be positive. But not all of them. Pseudomonas is usually negative, so that's okay. Other questions about inducible operons? Okay, well, the opposite is a repressible operon, one that is normally on until we turn it off. So the classic example of a repressible operon that we talk about in genetics and microbiology, some people call it the TRP, I call it the TRIP, it doesn't really matter. It is the operon that encodes the structural genes necessary to make the amino acid tryptophan. Okay, so cells need tryptophan because they need to make proteins, and tryptophan is an important amino acid, part of a lot of proteins. So normally this is on. Tryptophan isn't necessarily the most common dietary resource, I guess, for some of these organisms. So they typically have to make their own. The structure is very similar, except in this case, right, the repressor, the default status of the repressor, the default is inactive. It is not bound. This is the default that we're doing all this transcription because we need to make tryptophan. So under normal conditions, the E. coli are happily making tryptophan because the amount of tryptophan within the cell is pretty low. But maybe on Thanksgiving, they got a turkey and there's a lot of tryptophan. E. coli Thanksgiving, whatever. Now there's tryptophan. I don't need to make any more tryptophan. Remember, transcription and translation are expensive. If I have enough of the resource, why would I make more of the transcript and proteins necessary to make that resource? So when tryptophan levels are high, the amino acid itself, tryptophan, acts as what we call a co-repressor. It binds to the repressor protein. That's what they're trying to show here. And that causes it to become active. So the repressor bound to tryptophan binds to the operator and blocks transcription. So we call it a co-repressor. It doesn't directly repress, it binds to the repressor protein which represses. So neither inducers nor co-repressors interact with the DNA itself. Only the repressor protein does. And again, this example is the TRIP operon, but repressible operons work in this way. There will be a co-repressor that causes transcription to stop. And I have an animation. And this is the last animation I think that you have to watch today. There will be more.
The TRP operon of E. coli includes a promoter, an operator, and 5 structural genes encoding proteins involved in the biosynthesis of tryptophan. The TRP operon is a repressible operon, meaning that the genes are usually transcribed. When excess tryptophan is available to the cell, the operon is repressed, ending the production of the proteins of tryptophan biosynthesis. The E. coli chromosome contains a regulatory gene near the TRP operon. This gene is constantly transcribed and translated to produce repressor proteins. However, these repressor proteins are inactive unless excess tryptophan is available. Once activated by tryptophan, the repressor proteins can bind to the TRP operator, preventing RNA polymerase from moving beyond the promoter. When a repressor protein is bound, the genes for tryptophan biosynthesis cannot be transcribed into mRNA. When more tryptophan is available to the cell than the cell can use, some tryptophan molecules bind to and activate the repressor proteins. The repressor proteins bind to the operator, preventing the transcription of the structural genes that encode the proteins of tryptophan biosynthesis. This regulation prevents the cell from producing excess tryptophan when tryptophan is already available. When excess tryptophan is not available, the repressor proteins are inactive. The structural genes of the TRP operon are transcribed and translated, producing the proteins necessary for tryptophan biosynthesis.
Questions about repression. So again, these animations are not part of your text, but I did include them in the slides. So hopefully if you download the PowerPoint slides and you want to kind of review these concepts, you can always either rewatch them from that, or you can rewatch this part of the lecture video, either way. So I told you that the Lac Operon is not quite as simple as lactose present. Let's turn it on. I wish it was, because then I wouldn't have to talk about this part, which is a little bit more complicated. So it's a little bit more complicated because of course it is. Why would it be simple? It's life. Complicated. The Lac Operon is regulated by, well, really two different sugars. Yes, lactose. So that is not inaccurate. It is true to say that the Lac Operon will not be turned on unless there is lactose. You have to have lactose to turn on the Lac Operon. It's just a little bit simplified to say if there is lactose, it 100% will turn on. So it's just a little bit more complicated than that. And what is complicated is glucose. So think about it this way. When you were a kid, you had food set in front of you. May not have been your choice of food, but it was food. It was provided. And there were probably things on the plate that you liked. And there may have been vegetables that you didn't like. Mine was broccoli. It's weird. I like it now. I'm an adult. When I was like 6 or 7, ugh. So what did I eat first? Not the broccoli. It was not my preferred food source. Would I eat it eventually? Yes, because I would have been in trouble. Bacteria are the same way. They will eat because they want to survive. What will they eat first? Glucose, 100% of the time. Even if they have lactose. Right? Again, I'm talking about like a kid, right? If my mom gave me a plate that had a slice of chocolate cake and broccoli, duh, I'm going for the chocolate cake. That's my preferred food source. Eventually I'll eat the broccoli because my mom said I had to. So the cell will eat the lactose. It has to survive. It prefers the glucose. That's what we call positive regulation. The glucose is going to be consumed first. And that is regulated by a couple of different proteins. One is called CAP. Okay. And it's regulated also by a small molecule called CAMP. I think looking at the text isn't helpful. Let's look at a picture instead. So in a situation, you have a cell, right? So there's four possibilities for a cell. You could have high levels of glucose, and low levels of lactose. You could have high levels of glucose and high levels of lactose. You could have low levels of glucose and low levels of lactose. Or you could have low levels of glucose and high levels of lactose. Okay. So what happens is there's this little molecule called cyclic AMP. Cyclic AMP is usually at the highest levels when there is no glucose. So when there is glucose, we have low levels of cyclic AMP. Yes, glucose, low cyclic AMP. No glucose, higher cyclic AMP. So what cyclic AMP does is it activates a protein called CAP, catabolite active protein, or catabolite activator protein. And you see, right, we have glucose, but CAP isn't binding. So basically, if you have low levels of cyclic AMP, let's say CAP binding, you also have low levels of CAP binding, which implies if you have low levels of glucose, high levels of cyclic AMP, you have high levels of CAP binding. We're not even worried about the lactose yet. We already know what happens with lactose. Okay. So let's look at a condition now where we have a low level of glucose. So when we have a low level of glucose, what they're showing you here is that low glucose, you get high cyclic AMP. So cyclic AMP binds to this CAP protein. CAP becomes active, and now it binds to the promoter. It enhances the binding of RNA polymerase to the promoter. You really need CAP in order to get RNA polymerase to bind. So what we could think about is if we have low cyclic AMP, we have CAP binding. What about RNA polymerase binding? Okay, so if we have low CAP, we have low CAP, we probably also have low RNA polymerase. If we have high CAP, we have high CAP, we have high binding of RNA polymerase. And so right now, all I'm showing you is why when you have high glucose, you're not going to get a lot of transcription of the Lac Operon because you're not recruiting very much RNA polymerase. But the last thing to think about is the Lac repressor. Oops, I can't spell. Repressor. Okay. The Lac repressor has two options. It's either bound to the operator or not bound to the operator. If I have no lactose, right, the Lac repressor is bound. If I have lactose, the Lac repressor should be unbound. No lactose. Yes, lactose. But now we have to take these things together, and now we look at transcription levels. Okay. Well, here, my operator is blocked. It doesn't matter what else is going on, no transcription. Here, my operator is blocked. It doesn't matter what else is going on, I get no transcription. Now here are my questions. I have two situations where I have lactose. So in theory, I should get transcription. In practice, I do get transcription in both of these situations. But how much? So, I have no camp. I have no cap binding. Very little recruitment of RNA polymerase. My transcription level is going to be very low. There might be some that sneak through, but because I have glucose and I'm not synthesizing, this is a stress molecule. When I have glucose, I have no stress, right? I don't recruit RNA polymerase. Doesn't matter. The operator's free. Who cares? I have very little recruitment of RNA polymerase. I have very little transcription of that operon. But in a situation where I've recruited as much RNA polymerase as possible and my operator is free, now I can transcribe that Lac Operon. So it's the idea that the cell not only turns on the Lac Operon only in the presence of lactose, it turns on the Lac Operon at the highest levels when lactose is the only carbohydrate available. It doesn't waste its energy to consume lactose if the preferred carbon source is available. It prefers glucose. So full expression of the Lac Operon requires both, yes, the presence of lactose, but the absence of glucose. So how do we know this? How do we know that glucose is the preferred carbon source? There's a couple of ways. One, when you grow E. coli only in glucose, right? You see this nice, like, steep line. It grows pretty rapidly. When you grow E. coli only on lactose, the growth rate is much slower. It cannot grow as quickly when it's only given lactose. That's one. Okay. The other way that we know that glucose is the preferred carbon source is because we can grow E. coli and give them multiple carbon sources. So this is a growth curve of E. coli in media that contains both glucose and lactose. And what's really interesting about this growth curve, and when I find something interesting, you should probably find it interesting. What I find interesting about this growth curve is we see this nice log phase, nice rapid growth, and then we see a lag phase. And if we stopped right here, we'd say, oh, the interstationary phase, everything's fine. So how do I know it's not stationary phase? Because they don't die. They keep growing. But now they grow much slower. So they enter a second log phase. It's not as fast. The implication of just looking at the growth curve is that they have used up all the glucose. So they did their log phase. They used up all the glucose. Growth has paused while they start transcribing and translating the Lac Operon. Now all they have left is lactose. It's not their favorite. They'll use it. It just takes them longer. So that's just, me inferring things from the growth curve. But we also can test this at the molecular level. And what they show you here is that, well, you can do it a couple different ways. One, you look for the proteins of the Lac Operon. The really common enzyme is called beta-galactosidase. If you were to analyze what proteins is E. coli producing, here they do not produce beta-galactosidase. Here they do. Or you can look at the transcripts. You can look directly for the transcripts. Here there is no transcription of the Lac Operon. Here there is. So we know that they switch their gene regulation in the same tube. So this is an experiment that I know one of my colleagues at Fresno State does in a bacterial physiology class. You watch, they call it dioxic growth, 2 growth phases, dioxic. You watch them use their preferred carbon source, you watch them slow down, you watch them use the other carbon source. I inhaled the cake, I stare at the broccoli, I eat the broccoli. That is what we call catabolite repression, the presence of one preferred carbon source keeps production of this operon down until that preferred carbon source, the one that allows me to grow the fastest, is all used up. Then I can turn on the other genes because I don't want to die. It's just not my favorite. Okay. So dioxic growth curve, 2 logs and an additional lag. Before, eventually they'll go stationary and die, that's fine. You could continue it out that way. But those 2 log phases is really classic for switching gene regulation and utilizing a different carbon source. Questions about Lac, trip, and catabolite repression? Okay, bye, Leaf. See you on Thursday. And we'll pick up with mutations on Thursday.